Okay, everyone, we have made it to our last lecture of the semester. And this is where, again, I wish we were in person because some of the stuff we're going to talk about in this last lecture are really exciting and fun things to think about for those of you who are really looking forward to continuing in biology. And I just miss those last days of class with you all. We tend to laugh and talk, and I hope that the semester, all considered with all of its challenges, has gone okay for all of you. And I really do hope that I see many of you back on campus in the fall when we can again, learn like you're supposed to learn, which is together. So here is our last lecture for the semester. And this one is not going to be as packed full of information. It's a little bit more kind of, I don't want to use the word fun, but more fun, I guess, and a little bit more future looking with where we're going with the future, which is to me very interesting. So this slide is again one of the wonderful opportunities that I had over January to go to the Wellcome Collection, which is a museum of a lot of important things, and they had on display when we were there in January of 2020. They had on display the human genome sequence all printed out in books, and that's what you see here in all these white volumes with the numbers on them. That's the entire human genome printed out in books, and you can see the size of the font. It's a tiny, tiny little font, and there are thousands and thousands of pages. And so it's a really great visual to help you really realize and understand how large and complex the human genome sequence is. We've got 3.2 about billion bases, and so it was just a really interesting display. And so this topic that we're going to talk about now is really about genomics, and genomics again is that complete genetic composition of a cell or an organism. And so the human genome sequence is all laid out here in these multiple volumes of books. And so I want to talk first about how we determine the sequence. How do we determine the nucleotide order, the order of the nucleotides when we take on, either whether we're trying to sequence a small piece of DNA or a very large genome. So I want to first talk a little bit about the methods that are used to determine the order of the nucleotides. So again, DNA sequencing is the process that determines the order of the base sequences in DNA. And the main or primary method to sequence the human genome was what's called the Sanger sequencing method. And this was developed in 1977, and it is still widely used today. It's not, there are other methods we're going to talk about in a little bit here, but this method is still widely used, and Sanger received the Nobel Prize for coming up with this method, and it's what's often called the chain termination method. And so to understand how this works, we need to go back and remind ourselves of the structure of a nucleotide. So we've seen this many times this semester. So we have the triphosphate attached to the 5' carbon. This is a deoxyribose sugar. You look to the 2' carbon and we only see a hydrogen. We do not see a hydroxyl group. And then in this particular example here, a G base is attached to the 1' carbon. But what makes this what we call a dideoxynucleotide, right? Normal DNA is made up of deoxynucleotides, but this is a dideoxy. And what's circled here in red is you see there is no hydroxyl group on the 3' end either. And when we remember back, this is how we start to put all of these things that we've learned this semester together. When we think back to how DNA replication works, remember we synthesize the DNA in that 5' to 3' direction. And the phosphodiester bond is created by linking the 5' phosphate from one nucleotide to the 3' hydroxyl of the next nucleotide. When we use dideoxynucleotides, there is no 3' hydroxyl. And so when this happens, again, these nucleotides lack that 3' hydroxyl group, so synthesis is terminated when these nucleotides are inserted or incorporated into a growing strand of DNA. And that's the key to how Sanger sequencing or this dideoxy chain termination method actually works. So let's look at how we put this all together and how we use these dideoxynucleotides to determine the order of nucleotides in a DNA sequence. Okay, so this really outlines how DNA sequencing, or the Sanger method, actually works. So we are mixing together a whole bunch of different reagents, and really what we're doing is we're doing DNA replication in vitro. So similar to what we talked about with PCR, but there are a few differences here that allow us to determine the order of the nucleotides. So we have to have some sort of a target DNA or some sort of a template DNA, the DNA that we're actually sequencing, and that's shown at the top. So you can take a piece of DNA and you can clone it into a recombinant vector and make a recombinant molecule, which we talked about in the last set of slides. You can also do this with DNA that's not in a vector, but nonetheless, you need some sort of target DNA. This is the DNA that you're actually sequencing. And remember when we talked about DNA replication, the polymerase needed a primer, so you need to include a primer. Primers are a part of this, whether you're talking about sequencing, DNA replication, PCR, primers are part of it. So you have to include a primer. We need to include the actual nucleotides. Right? If we're going to synthesize DNA, we need those building blocks. So you put in some normal deoxynucleotides, right? Those that have the 3' hydroxyl group on them. You include the DNA polymerase, which is the enzyme then that synthesizes the DNA. And then in small little ratios, you also include those dideoxynucleotides, or those DDNTPs that I showed on the previous slide. And remember, whenever those get incorporated into a growing chain, the chain stops. It doesn't grow any longer. No more nucleotides can be incorporated in the end. And those dideoxynucleotides are actually labeled with different colors. So if we look at this slide, we see our target sequence at the top, and then we have all of these ingredients I just laid out go into a tube. And in this case, you can see that the DDG, is labeled with a yellow fluorescent tag. So you actually chemically synthesize the dideoxynucleotides. In this case, will have a yellow fluorescent tag on them. The T's appear to have a red tag, the C's blue, and the A's are green. So in a test tube, we start to copy or replicate the template DNA. And occasionally, one of these dideoxynucleotides will get incorporated into the synthesis of this newly made piece of DNA. And you set up the ratios so that it happens randomly, but it happens enough so that there is chain termination at every nucleotide along the way. So you see all these different fragments of DNA being synthesized, each having a different dideoxy terminating the end of that. And so then you can take these different sized fragments, and remember we talked about gel electrophoresis many times this semester. You can run these different fragments on a gel and separate them out by size. So the smallest one, if I look at that picture, the very smallest fragment is the one that just has the dideoxy C nucleotide. It's literally 1 nucleotide long. Then the next one is 2 nucleotides. I'm going from the bottom up here, and the next one is 3, and so on. So if I run those on a gel, I will separate them out by size. And the smallest will run the fastest, migrate the fastest through the gel, and the larger ones will migrate slower. And remember, each one of these fragments is labeled with the dideoxy color that is on the end. And if each of them is only 1 nucleotide different in length, I can read the colors from the bottom up and determine the sequence of the piece of DNA, right? So this tells me that the sequence from the bottom up, from the smallest fragments to the largest fragments, must be CAC, CG. Again, I'm reading that sequence in the diagram on the top right, because I can look at the colors of those fragments that have been separated on the gel. And then there's a laser reader that reads those outputs, and you get sort of this peak diagram that's shown on the bottom right there. So this is the readout that you get from doing this kind of sequencing. So whenever a blue band is detected, you get a blue peak. And so you get these kind of outputs, and from that you can determine the sequence of the DNA in that template that you were analyzing. And again, this was basically the method that was used to sequence the human genome, over 3.2 billion bases, and that whole project was completed back in 2003. And so we think about that human genome sequence that we obtained back in 2003 as what we might call the reference human genome sequence. It was not the sequence of an individual person. It was actually template various sequences from a number of different people. So it is not the sequence of one human. It's kind of a composite, but we still consider that to be kind of the reference human genome sequence. And so again, this was sort of the state of the art back in the early 2000s, but since then, there has been what we might consider a revolution in DNA sequencing and genomics. And so let's talk about why and what that looks like in the next few slides. In this graph, I'm showing you the cost, right? All these, sequencing 3.2 billion base pairs was not, cheap. The reagents cost money. People doing it paid them to do it, and it was actually, quite a bit of money to sequence the human genome. We're talking, this is on the y-axis here, we have cost in thousands of US dollars. So we're talking on the order of billions of dollars back in 2003 when the first human genome sequence was obtained. But what you can notice by looking at the green line here, we see all of a sudden a very precipitous drop. Long about, 2008, we see the cost of sequencing a DNA drop dramatically. And again, you see that compared to Moore's law, which is some law that people have put together that predicts how computing costs would go down. And so it dropped much more than Moore's law predicted. And you can see this graph goes out to 2013. Clearly we're at 2020 right now, and currently one could sequence the entire human genome for way less than $1,000. And a lot of people have had their genome sequenced, and so it's becoming very, very inexpensive. And so I want to take a little bit of time to talk about why that is. What happened in the DNA sequencing area that allowed for the cost to drop so dramatically, which has, again, revolutionized how we look at sequencing human genomes. Costs are not a big consideration anymore. It's relatively cheap and quick and efficient to sequence lots and lots of DNA. So what developed were sequencing techniques called next generation DNA sequencing. And some of these are fairly complex, and there's lots of different platforms that DNA sequences are lots of different platforms that allow for DNA sequencing. And for those of you who are really interested in a lot of this, I teach a 400 level molecular genetics class. Hopefully when you get to be juniors or seniors, maybe I'll see some of you in that class, because we do talk about this particular topic in much more detail in that class. So next generation sequencing, lots of different platforms, but let me just give you just an overview, a basic overview of what one of the platforms, the most commonly used platform, looks like, so that you can begin to understand the enormity of the data that's collected and how a lot of this gets put together. So this is a really simple example to help hopefully illustrate how some of these next generation sequencing platforms work. So if you recall from when we were talking about Sanger sequencing, you have your template DNA cloned into a plasmid vector, and then you sequence it in the solution, and you mix all these reagents together. Well, next generation sequencing, almost all the platforms, rely on attaching your template DNA to some sort of solid support. And so that's what you see here. So we see that gray sort of slide and those little, ball-like structures that are attached. So the DNA is physically attached. It's not just floating around in a tube. And again, a lot of nanotechnologies are used to allow for different DNA fragments to be attached to various locations on a slide. And you see one of the flow cells, as they call them, that are used for this type of sequencing in the upper right there. And so you get an idea. It just basically looks like a microscope slide. So if we zoom in on this microscope slide, what we would see are all kinds of little pieces of DNA that are physically attached to the surface of this slide. And there are literally millions of them attached to the surface of the slide. So then what you do is you perform your sequencing kind of one nucleotide at a time on these little areas where your template DNA is attached. And so if we look at, again, I'm very much oversimplifying this, but let's just look at the frame where it says cycle 1. So I'm zooming in on a small spot on the flow cell where I have 2 template DNA attached to the flow cell. And so the first base gets added to those millions of template fragments that are on the flow cell, and a little fluorescent light is emitted when the new nucleotide gets added to the growing end. And so in cycle 1 there, I see a green fluorescent spot at one little location, and I see a yellow or an orange fluorescent spot at that other template DNA. So then I continue and I add the next nucleotide. And in this case, because of the sequence of the template DNA, I see 2 red spots. And then for cycle 3, I add the next nucleotide, and so on and so on. And so you put this into these fairly complex machines that are able then to read the color that's emitted at every single cycle for millions of spots. And what you see in the bottom right there is sort of an example of what this might look like. So remember, every one of those tiny, tiny little colored dots represents a piece of template DNA that's physically attached to the flow cell. And what this diagram is showing you, you can see with the inset, is that this is what about 20 microns or 20 micrometers across that flow cell would look like. And what does it say? 250 million clusters or 250 million template DNAs are sequenced or are assessed or assayed per flow cell. And there are some machines now that have ramped this up where you literally can get billions, I'm talking about billions of sequence from one single run, which takes a few hours. So what took years and years and many hours of manpower back in the early 2000s can take literally 6 hours plus now. Because again, you can, on one of these machines, you can actually sequence up to the equivalent of about 10 human genomes in one single run of the machine that might take a few hours. So this is what led to that graph that I showed on the previous slide. This is what led to that precipitous drop, because it's very inexpensive, very rapid, and easy to use these machines now that allow you to sequence. I mean, you can sequence one human genome, the equivalent of one human genome, easily in a few hours now. And again, it took years and billions of dollars, and now it takes hours and just a fraction of the cost. So this is where we are with DNA sequencing now. And so DNA sequencing has really become a revolutionized, and it is to the point where probably any of you, if you wanted to, you could go have your entire genome sequenced if you wanted to. So what I want to do next is talk a little bit about what we've learned from a lot of the sequence that we've obtained, not only with the human genome, but also by sequencing other genomes. We've sequenced lots of bacterial genomes, lots of fungal genomes, humans, other mammals, other animals, so we'll talk about what have we learned since we've obtained a lot of this sequence data. One thing we have certainly learned by sequencing a number of different genomes is that the size of genomes across various eukaryotes is highly varied. When we compare that to genomes in most prokaryotes, Most prokaryotes, the genome sizes stay within a fairly reasonable range, but we do note that many of the nuclear genomes among eukaryotes are widely varied in size, and it's not necessarily related to the complexity of the organism, as you can see in this graph. So we're looking at a number of different eukaryotic groups here, and you can see how many nucleotides there are per haploid genome. And what you notice is there's a very broad range in plants. Certainly amphibians have a very broad range, but you can see mammals, it's fairly narrow as far as the number goes, and we certainly are not at the high end. And what you're looking at here are two plants that appear to be fairly related to one another, but their genome sizes vary quite a bit. I mean, we're talking about, this is a factor of 10 on the x-axis at the bottom. So we did learn that the genome sizes vary, and that size is not necessarily related to the complexity of the organism. And again, I'm going over some of the highlights. We're not going to get into the details, but a couple of or at least one reason for these varied genome sizes is something we discovered that is true in the human genome also. So let's look at that in the next slide. So we certainly have learned in the human genome that there are a number of gene families. So if we follow this particular graph and start at the bottom, okay, and we've got on the y-axis here, we have a prediction of how many millions of years it has taken for this kind of genome evolution to take place. So we know in the human genome that there are numerous globin genes. I don't know if we think back to when we started talking about gene regulation, there was a slide that I showed you that had the different globin genes that were expressed during different times of development, right? There were some that were expressed early in fetal development, and then they got turned on and off, and then there are only a couple of them that are expressed in adults. And so those are the genes that we're talking about here. Those are the globin genes that we're talking about here. And again, they were indicated in that slide I showed way back with various Greek letters. And so what we know and understand as we've looked at the human genome sequence is some of these sort of related, or we often use the word homologous. Homologous sequences are those that share common orders of nucleotides. What we've learned is that there are gene families within species. And many of these likely arose during evolution as sequences randomly duplicate, and there are mechanisms for this that we're not going to get into. But if we start at the bottom, maybe once upon a time there was this ancestral globin gene, and then a duplication event occurred, right? So now instead of having one globin gene, there were two in the genome. And then over time, through natural selection and other types of diversification, one of them became what we now know to be our myoglobin gene. And then the others, maybe hemoglobin genes, then duplicated again to give us what we now know as the family of what we call alpha globins and the family of beta globins. And then those duplicated even further to give you all of those different globin genes that are shown there at the top on chromosome 16, and all of the various beta globin genes that are shown at the top there on chromosome 11. But if we look at all of these genes, whether we're talking about those on chromosome 22, the alpha globin genes on 16, or the beta globin genes on 11, if we compare them, they're very related in sequence, and we call these paralogs. So any sequence that shares something in common in general is called a homolog, but gene families within species, shared sequences within species, are called paralogs. And there are lots of examples of these in the human genome and in other organisms as well. And not only is it an interesting way to try to understand molecular evolution, but it also helps us understand genes that are related in sequence and also somewhat related in function, right? Because we know all of these that are listed here do play some role in the oxygen carrying capacity of cells. So paralogs are these gene families within species. And as we've sequenced many, many organisms, we not only can compare sequences within a genome sequence, we can compare sequences between species and get some information about related genes and, again, evolution and speciation and things like that. And so what's shown here, we're still talking about those globin genes, but what we're looking at here is musculus is mice, and then there's a rat species shown here as well. And we can also follow related sequences between these two species. So we're looking here particularly at beta globin genes, and we can compare the beta globin genes that we see in the genome of mice to the beta globin genes that we see in the genomes of rats. And at the top, you can see that comparison. So we're comparing the sequences and looking for how related those two DNA sequences are between mouse and rat beta globin. And related sequences that we see between species are what we call orthologs. So again, anything related in sequence is called a homolog. If it's related sequence within the species, those are called paralogs, and related sequences between different species are called orthologs. And again, this has given us some insight into how species have evolved and diverged from one another, and what some of the sequences are that they share, and some insight into how that diversification or change over time has occurred. And on the next slide, we'll talk about one that's of particular interest to the human genome sequencing. So in this slide, again, this was back in 2010. So when we talked about PCR way back when, I'm trying to bring a lot of things together at this point in the semester. And when we talked about PCR way back when, and we were talking about how you can use PCR to amplify ancient DNA. Well, that's exactly what was done here. Some Neanderthal bones were found, and DNA was extracted from those bones, PCR amplified, and then the DNA sequence of some regions of Neanderthal humans was actually determined. And so this was a big publication in this journal Science, again, back in 2010. And what we were then able to do is to compare some of that sequence to present day humans. And what you see here in the graph, again, this is not throughout the entire genome. I believe this was comparing some sequences within the mitochondrial genome. So remember, mitochondria have DNA as well. And so they were looking at the differences between various regions in the mitochondrial DNA. And so what you see when you compare human to human, we'll talk about this some more, but we certainly know if I compared my mitochondrial DNA sequence to yours, we would see a number of differences. And that sort of outlined the range of numbers of differences is shown there in that first grouping when you compare one human to the next. But then you can see when we compared present-day humans to the information we got from the Neanderthal genome, we see that those differences, there are quite a few more differences when we compare certain regions of the mitochondrial DNA. So that entire group, that entire range is shifted towards the right, so there's more differences. And then to illustrate it, or to show the other extreme, if we compare the human DNA sequence to the chimpanzee sequence, we of course see a larger number, or that entire range is shifted even more to the right. So again, interesting information can be gleaned by comparing sequences, not only within a particular species or within an organism, but also comparing them between organisms. And you can get interesting insight then as to where and how they diverged from one another, and interesting insight into genes that are shared between different species. And so there's one last important thing that came out of, has come out of all of this genome sequencing. And so I want to spend the last few slides just highlighting that. So if we get nothing else out of what we are able to do now with this whole genomic revolution, it's this idea that we can now very easily look at what are called single nucleotide polymorphisms, in humans particularly. And sometimes these are just referred to as SNPs. So a polymorphism, morphism means shape and poly means many, and single nucleotides. So these are single nucleotide differences that are across all humans. So again, with the ability to sequence the human genome for very low cost and very rapidly. I mean, now I don't even know what the number is. Once upon a time, again, back in 2003, we knew the sequence of a single, composite reference human genome. Now, I mean, there are thousands, probably hundreds of thousands of human genomes that have been sequenced. And so what that allows us to do is to compare one human genome sequence or one area of the genome to another. And what we've found is if you just randomly pluck two humans off the street and you sequence their human genome, what you will see is that they are different at about 3 million bases. And these are single nucleotide differences that are present between humans. And again, when you're talking about 3.2 billion bases, having 3 million differences is not a significant amount of sequence that's different. But you see this human genetic variation. And so each one of us, at every generation, so when you were born and your parents' egg and sperm came together, you now have about 100 new variations, 100 new SNPs or single nucleotide changes that are just unique to you, right? They weren't present in your mom, they weren't present in your dad, they're not present in your siblings, but at every generation there are about 100 of these SNPs that occur. And so again, what we've been able to do then is use this human genetic variation to learn and understand a lot of things about what is personal in a person's genome, and probably what many of you are familiar with. This has also resulted in this huge boom of direct-to-consumer genetic testing through 23andMe, through Ancestry, and all of those, and really being able to quickly determine a few highlighted SNPs or polymorphisms in an individual's genome has really allowed for a number of different things. And I just want to highlight a couple of them. So first of all, it's allowed we can use SNPs to tell us something about ancestry, and that is what's used in some of these direct-to-consumer genetic testing. It's allowed us to map genes that are involved in some complex traits and diseases, and I'll show you a slide on that in a minute. And then it also has really led the way to a more personalized approach to medicine and healthcare and treatments that can be tailored specifically to a person's basic genetic profile or their SNP profile. All right, so let's look at each of these quickly in the next couple slides. So again, these single nucleotide polymorphisms occur, and as you might imagine, again, as I said, if I pluck two random people off the street, you're going to see a number of different variants. But certainly people who are more closely related are going to share some of those variants more than two people who are unrelated. So it's hard to read on the bottom, but this is a graph showing the amount of related, how many SNPs in general are people share. On the bottom are people of various countries. I think there's people of African nations, there's European nations, but all of that is across the bottom. And what they're doing here is they're looking at 4 of these polymorphisms. And one of them is in red, one's in blue, one's in yellow, and one's in purple. So they're analyzing people from these different regions and looking for what percentage of them actually share a particular SNP. So let's just follow the blue line as an example here. So I'm looking at the sequence of people from all of those different regions that are listed on the bottom there across the x-axis. And what I see, if I follow the blue region, is that there's this particular SNP that I'm following seems to be highly shared, most commonly similar amongst people who are from all of those nations that are listed in about the middle of that x-axis. If I look at the red line, that's looking at a different SNP, and I see that that one seems to be shared among people, most highly shared among people who are from those regions that are at the front of that, or the beginning of that x-axis. And then as I go down the x-axis and come across people who are from those countries listed more towards the end, I see a much higher coincidence of that SNP in those individuals. And then at the very end of the x-axis is where I see an increase in the common shared SNP there shown in purple. And so this is really what a lot of those direct-to-consumer testing centers do. They take DNA, they analyze the SNP profile, what variant, what human genetic variant do people have in certain regions of their genome. They don't necessarily sequence the entire genome. And they have collected data, lots of data over the years to say, well, if you have the profile that's indicated, or you carry the SNP that's indicated there in red, we can say you have some ancestry or some ancestors that are from some of those countries where other people share or have that same variant. And so, again, using these SNPs as a way to follow ancestry, that has been one big advent in some of the human genome sequence, and being able to obtain sequence so readily and so inexpensively. So I know that this slide is really hard to see, and I think that's intentional. I just wanted you to see sort of the enormity of all of this. So way back when, we did talk about complex traits. If you remember complex traits and complex diseases, these are those traits and diseases that are caused or influenced by a whole slew of genes. And I think, we talked about human height, we talked about the pigment of skin color, and we were talking about, what if it's influenced by 3 or 4 genes? Well, what about some of these things that are literally influenced by the alleles at, 10, 12 genes? How do we ever wrap our heads around that? How do we ever get at the genetics behind that? Well, being able to look at human genetic variation has allowed us to start putting some of this together. So again, I'm oversimplifying, but what you can see here, if you look carefully, is these are all the human chromosomes, 1 through 22. Sorry guys, the little Y chromosome there is shown at the bottom, and there isn't a lot of contribution to complex traits and diseases that are on the Y chromosome. But all the other chromosomes are indicated here. And what all these colored dots show, and the legend is there on the bottom right, but what they show is through analysis of all of this genetic variation, and how that's actually done is fairly complicated. And like I said, we do go into this in much more detail in my upper division class. But all those little colored dots indicate a gene or an area on a chromosome that contributes in some way to the disorder or the trait that's listed there in the legend on the right. And again, you can see some of these dots show up multiple times scattered throughout the genome. So being able to follow and analyze this human genetic variation has really allowed us to get a better handle on what parts of the genome and what different genes, again, many, many different genes that are involved in determining some of these complex traits like cardiovascular disease. We're talking about body mass index. We're talking about, autoimmune diseases, cancer, all of these things that are very complex and probably influenced by many, many different genes. And then the very last thing I want to leave you with is how the human genome sequence and analyzing this human genetic variation is really leading us towards what we might refer to as personalized medicine. And I have no doubt it's already starting, that in the future, and certainly in your lifetimes, what we will see when we go to see our healthcare providers, It's not a one size fits all anymore. And so if we look at this diagram where we have all the colored people, the current strategy and the old strategy is, we kind of treat everyone the same. And so if you come in and have a particular ailment, whether that be cancer or, something that you need some medication prescribed for, I mean, everyone sort of gets that same therapy. But in the future, the idea is there will be some sort of a genetic analysis, because as we've gathered data, maybe we know that this particular person with this particular set of SNPs and this particular genetic profile responds really well to this particular cancer treatment. And people with this genetic profile, well, maybe they have the same breast cancer, but we've learned that if they have this set of profile, that they don't respond as well to that treatment, and they maybe respond better to a different type of treatment, a different type of cancer treatment, a different type of drug regimen, whatever the case may be. But that's really what we're starting to talk about here with personalized medicine. And so you see how this all sort of fits together in that diagram there on the right. So you've got personal genomics, because it's so easy to obtain, the particular variation, the particular variant profile of individuals. That's very cheap, to the point where you'll come into a doctor's office, maybe give them a drop of blood, they throw it into a machine really quickly and can determine important areas across your genome that have particular variants. And that might dictate how your particular ailment is treated. That may dictate which drugs you're prescribed, or which regimen of treatment that you're on. And this is the future. And this is, again, based on the advent of being able to readily sequence and understand genomes at a level that we were never able to understand before.